Hi everyone, and welcome back to the second part of the medicinal chemistry crash course with Matthew Perry. Without further ado, I will hand over the floor to you, Matthew. Thanks a lot for joining us again.
Hello again, everyone. Welcome to part 2 of the medicinal chemistry crash course. Today, I'm going to talk about some fundamental concepts you need to understand for medicinal chemistry, so drug metabolism, drug safety, and then some basic principles that are used in medicinal chemistry. And I'll go on to talk about the lead identification phase of the project. So, without further ado, let's get into the talk. To start this section, I want to begin with a brief introduction to pharmacokinetics. It's absolutely essential for medicinal chemistry to understand pharmacokinetics, at least in broad terms. So pharmacokinetics can be defined as what the body does to a drug. We need to know how our drug will behave in a patient. We obviously can't test everything in patients, so we need to have to predict that. What we can test is we can use human enzymes and cells, so microsomes and hepatocytes. We can do experiments with animal enzymes and cells, and also experiments in animals. And we can use the data we generate from those experiments to predict the human pharmacokinetics from a combination of them if the hepatic metabolism is the major route of elimination. So, pharmacokinetics. The way that pharmacokinetic experiments are done, they are typically dosed first as an intravenous bolus dose and then orally. And I'm assuming here that you're looking at an oral treatment. If you're looking for a different route of administration, then you will have to have appropriate pharmacokinetics for that. But given that oral drugs are the most common, I'll focus on that here. And so these experiments generate profiles showing the concentration of drug over time. So you'll get a curve that looks like this. This is a typical IV administration profile. You've got a concentration here. It's typically logged because it drops very rapidly. It will often classically have two phases, a sort of initial distribution phase. So you administer it as a bolus, goes into the systemic circulation, very high concentration, and it distributes into tissue. And then you reach this point where it's distributed into the tissues and you start seeing elimination by metabolism and excretion of your compound. And so you get this effect with time over dose. And these experiments will generate four main numbers for you. From an intravenous experiment, you will get a half-life, and that can be measured from this elimination phase, and that's the time it takes the body to eliminate half the dose. So you can measure as you drop down here, you will see that after a certain period of time, you have half the concentration that you had before that, and then the same period again, it will be down to a quarter and so on. There is another number that you generate, the clearance, which is the rate at which the drug is removed from the plasma. And the third number that you generate is the volume of distribution, which is, it's a notional figure. It's not a real volume, but it represents the partitioning into tissue. So a drug that has a very small volume of distribution, i.e. that's only in the plasma, will have a volume of distribution that corresponds to that of the plasma, whereas one that distributes extensively into cells, membranes, whatever, that will appear to have a much larger volume. And so by understanding the volume of distribution, you can have an idea of what's happening to your drug. An oral experiment, so in an oral experiment, the compound is given oral PO per os, but often by sort of oral gavage into the gastrointestinal tract, and you will get curves that look like this. This is a repeat dosing experiment. Typically early on, you'll just do a single administration. So what you see in your experiment is that the concentration increases with time up to some point where you reach Tmax, and it will then decay away again over time again. And now from this point on, it will be roughly parallel, you hope, with your IV administration, and you will enter the same elimination phase because it will be the same processes acting upon the material that's been absorbed in whether it's been administered IV or orally. And you can work out from this curve and from the area how much of your compound got into the systemic circulation, and that enables you to calculate what the bioavailability of your drug is. That is to say, how much of the dose you give actually makes it into the systemic circulation. And you can see here in this curve, actually this isn't a logged curve, and you can see here in this one the person who's drawn this curve up has marked on the half-life, and so you can see the concentration drops from 50 to 25, and you can see that at the same another period of that half-life later, it's gone down to 12.5. Drug metabolism. Let's think about what happens to an oral drug. The drug is taken, let's say it's a pill, it's taken into the stomach, into the gastrointestinal tract. At some point there, it has to dissolve. You will be giving it as a solid. That dissolution rate will depend upon both the kinetic and the thermodynamic solubility. It also depends on the pH. The stomach is, as you know, fairly acidic, and then as the drug progresses, it goes into anything absorbed, goes to the small intestine, the large intestine, and then is excreted in the feces. But when you go from the stomach, which is very acidic, to the small intestine, the pH changes quite drastically. Most drugs are absorbed predominantly from the small intestine. It is typically the neutral species that's absorbed. So for an acidic drug, that will already have an impact because it will be predominantly the anion that's present, given the pH of the small intestine. Nevertheless, the surface area of the small intestine is much larger than the surface area of the stomach. So although you have a lot more neutral of an acid, it's still predominantly absorbed in the small intestine. And typically there is some absorption from the large intestine, but the most important is the small intestine. Drug that's absorbed goes from the small intestine, goes through the portal vein to the liver. That's important. The liver is the first port of call. So that means that any metabolism that happens in the liver can happen before your drug has been exposed to anything else. The blood then goes round from the liver through the systemic circulation to the heart, to the lungs, round, and then to tissues, to the kidney, and to the liver. You will get a certain amount of excretion of drugs, or at least you can do, from the kidney. Drugs can go from the bladder into the bladder and then into the urine. That's highly dependent upon the physicochemical properties of the drugs. Most drugs are metabolized predominantly in the liver, and they end up being excreted typically through the bile and going back into the small intestine. Now this can mean that if it's excreted unchanged, you can actually get it then reabsorbed. Normally, absorption is by passive permeability. However, there are transporters in the gut that are there to take up specific nutrients that the body needs, for example, amino acids, and it is possible to either have a drug that is taken up by active transport or modify it in some way so that it is taken up by active transport. There are also specific transporter proteins in the gut wall that are there to get rid of things that the body doesn't want, so-called efflux transporters. If your drug molecule is a substrate for efflux transporters, you may find that although it's quite permeable, it gets kicked out again very quickly. The distribution, I've talked about this. Again, the distribution is quite dependent upon the ionic state, Acids are very highly plasma protein bound, and so they're predominantly found in plasma, and this translates into a low volume of distribution. Bases, on the other hand, tend to have a very strong affinity for lipid membranes and also for acidic organelles like lysosomes. They typically have high volumes of distribution. Neutral molecules tend to be somewhat intermediate in terms of volume of distribution, but they are distributed quite widely beyond plasma. Now, that's the route, but there's also metabolism going on. Now, metabolism can occur in the gut, but principally happens in the liver. It can happen in the plasma. It can happen in other tissues as well. And again, principally, metabolism is by enzymes. And the major metabolic enzymes are split into two classes. There are what are known as primary metabolism. These are oxidative enzymes, cytochrome P450s, and there are 5 major isoforms that are involved in the metabolism of drugs: CYP3A4, 2C9, 2C19, 2D6 and 1A2, of which CYP3A4 is the most frequent metabolizer of drugs. And these are heme enzymes. They typically add oxygen to drugs. So they do reactions like hydroxylations and epoxidations. But the initial adducts can react further, so that's not necessarily what you see. There are other metabolic enzymes, aldehyde oxidase, uridine glucuronosyl transferases, monooxygenases. They are important in specific cases, but they have a narrower substrate range than the cytochromes, which basically will metabolize anything. And then there's secondary metabolism, which is conjugation, which is the addition of groups that make molecules more soluble, so glucuronic acid or sulfates typically. And these are secondary because they usually happen subsequent to some kind of oxidative metabolism, putting on a handle that can then be conjugated. If the drug starts out with a conjugative handle already on it, for example, a phenol, then secondary metabolism can happen directly. And the role of metabolic enzymes is to make any kind of xenobiotic, which includes your drugs, but also any poisonous things you've ingested, more soluble and therefore able to be excreted and got rid of from the system. So excretion into feces and urine. Very hydrophilic drugs, so like drugs with less than 0, will be excreted into urine unchanged. So some drugs will get excreted unchanged, but typically a drug gets metabolized and then excreted. So a couple of examples of drug metabolism. So sildenafil, probably better known to most people as Viagra, is predominantly metabolized by cytochrome P450-3A4. And this happens, I said this is an oxidative metabolism. It adds a hydroxyl to this methyl group on the piperazine ring, but that's unstable. And so formaldehyde is lost and you get this more soluble major metabolite with the secondary amine. Paracetamol, in turn, another well-known drug, that's a phenol, so you can get directly sulfation and glucuronidation, so secondary metabolites forming of this. There is also oxidative metabolism through P450s, which can either give this dihydroxy analog, but you can also form this quinone imine. And the quinone imine is a very important metabolite of paracetamol because that is a Michael acceptor. It reacts with glutathione. And glutathione is an important component in the body that's there to take out reactive species like this quinone imine, so it reacts with that, detoxifies it, and also makes it water soluble. And the toxicity of paracetamol arises whereby if you generate large amounts of this imine, you will deplete the stores of glutathione and then start seeing the toxic effects from this compound here. Moving from metabolism, I'd like now to talk about dose prediction. So this is something that will become quite important to the project during the lead identification phase and even more so during lead optimization. So I've already said that ideally an oral project will be aiming for once daily administration, but you might be going for twice daily. The question then you face is how much compound do you need to give to your patient? Because you want to keep that within a reasonable number, a reasonable mass. And so the purpose of doing this then is to identify how you need to optimize your compound in order to get to an acceptable dose. And so that will influence the design choices that you make in order to identify which properties you need to improve in order to get to your desired target profile. So the principles of this apply whatever means of measuring potency you're using. For the example I'll give here, I'll assume that you're working with average potency, so the average potency over your dosing interval. It could be that you're working with the maximum or the minimum, but in general the principles remain the same. So you can calculate the human dose based upon knowing what the average concentration you need to achieve at steady state. So that's after a few days dosing when the concentration levels have reached a steady state. And this is obviously the unbound average concentration. And then it's also affected by the intrinsic clearance, by the dosing intervals once or twice daily, and by the fraction absorbed. And the value of looking at this is this can translate into properties that you can actually measure in your screening cascade, so relatively early. So the average concentration that you need to achieve depends upon the potency of your compound in a cell assay you might typically. So if you know what that is, then you can calculate what the average concentration you need to achieve is to get an effect. Likewise, the fraction absorbed depends upon the solubility and the permeability of the compound, because the closer to 100% absorbed of your dose, the less you need to give. And then finally, the intrinsic clearance. You can calculate what the intrinsic clearance in the liver will be from measurements using hepatocytes and microsomes. And you can validate that. You'll have clearance data for human hepatocytes and microsomes, but you'll also have it for animal species. You can see what happens when you take your compound in vivo in an animal. Does it predict? If it predicts well in animals, then you've got confidence that it will predict well into patients. And so using this type of equation enables you to calculate what the dose you'll need to give to a patient is, and therefore how close you are to reach your target profile for your compound. Okay, so as an example of the application of dose prediction, I'll use Pfizer's SGLT2 inhibitor program. So they'd worked on this for a while and they got this early lead, which looked quite a nice compound, a 7 nanomolar potency. The rat PK didn't look too bad. Volume of distribution around 2 litres per kilogram, half-life of about 2 hours, plasma clearance of 22. So they went away, they measured the human metabolism, both in microsomes and hepatocytes, and then they used a combination of these to predict the human PK. So you get a plasma clearance of about 4.6 milliliters per minute per kilogram, volume distribution about 2, half-life was less than 5 hours. Now these values are all ranges, they're not precise numbers because there are errors in them. And the consequence of these was that the dose they got, the dose prediction, was above 100 milligrams, and that was higher than they wanted. And worse than that, it was very heavily dependent upon this half-life prediction. And if it came out at the lower end of the range of possible numbers, this dose would go up very considerably. And a big part of the problem was that the hepatocyte clearance was very high, and that was due to phase 2 metabolism, so conjugation, I think, as a glucuronide. So rather than take this compound forward, they did some further medicinal chemistry, they changed the scaffold a bit, and they came up with this optimized compound. And now this is significantly better. So you can see they've reduced the rat clearance, although the volume of distribution is also reduced, but the half-life is now twice as long in rats. The human microsome clearance is about the same, but the hepatocytes have come down, so it's not being subject to the same kinds of phase 2 metabolism. And so they predicted a lower clearance in patients. About a third of they had their volume of distribution was predicted to be about the same, but that now resulted in a 12-hour half-life and a greatly reduced predicted dose of 4 milligrams. So why is this? Well, they've increased the potency by nearly tenfold, so that obviously is going to reduce your dose. It's got a longer half-life, and that also not only reduces the amount of material that's needed, it also reduces that risk that we had here, or they had there, with the short half-life. And so they got this much lower dose. I mean, that's very attractive. And so this compound was taken into patients, and this is what they got when the values were measured. So this intravenous study was carried out in healthy volunteers in a phase 1 study, and I mean, that was really very close to the predicted value of 12 hours, 13 and a half hours. When they went orally into patients in a slightly later phase, the half-life's a bit longer still. And then these are the doses that the compound is actually now on the market at, so 5 or 15 milligrams. So you can see that prediction was actually pretty good. So really nice example of the application of dose prediction. Drug safety. I said at the beginning that safety is very important. Your drug must not cause undesired effects, and drugs can be marketed, but they can be restricted in use. It's what's known as having a black box warning. They have something on their prescribing information as a big black box warning. The doctor is going to prescribe it, danger, and that will affect the sales. If the side effects turn out to be really serious, drugs can and are withdrawn from sale. So you've spent all that time and money getting your drug launched onto the market, and then, oh dear, it's causing serious problems. You're not going to be able to sell it anymore, and you might be liable indeed for damages from people who have suffered serious side effects. So very, very important. On-target toxicity. These are unwanted effects that you get from whatever it is you're doing to the target. And so this is something that your project team are going to try and understand the consequences of through screening in cells, through animal studies. Off-target toxicity, on the other hand, is consequences of effects that you might have on other targets. And so as you go through the discovery phase, you will need to profile your leading compounds in screening panels. You will try to understand what else it affects and how it affects it. How potent is it at these targets? Are there ways that we can do it? And you may need to set up assays to measure that activity on these important off-targets and add them into your screening cascade. And then also you need to consider what is an acceptable safety risk for your potential product. So think about, who are your patients? How old are they? How seriously ill are they? You can imagine that a drug to treat cancer in older patients who are at risk of dying potentially quite rapidly can afford to have a bit more of a toxic side effect than would be tolerable for a drug treating asthma in children, for example. I want now to talk about some key concepts. I've mentioned lipophilicity a little bit. Lipophilicity is a very important concept to understand in drug discovery, in medicinal chemistry. Lipophilicity is the tendency of a drug to partition into a hydrophobic medium. It's classically measured between pH 7.4 buffer and 1 octanol. So pH 7.4 buffer is used as a model of physiological fluids, and 1 octanol is a model of lipid membranes. The simplest form is log P, which is the log of the concentration of the drug in octanol over the concentration of drug in buffer. However, if your drug ionizes, then you need to modify your concentration by including the ionized form in your calculation. And this will give you two numbers, either the log P or the log D. Many properties are influenced by lipophilicity. And there's a plot here from a project I was involved with a few years ago, whereby metabolic stability was essentially inversely correlated with lipophilicity. So as the compounds became more lipophilic, the stability measured here in this log of the intrinsic clearance became lower. And for this project, the problem was that the potency went in the opposite direction. So as we made the compounds more lipophilic, the potency went up. This was a program targeting CCR inhibition of CCR3. And basically in this project, we were never able to get away from this simple lipophilic driver. And that then was a reason we stopped that program. I say potency. It's not just the potency at your target that can be affected by lipophilicity, but often activity at off targets can be affected. So what you will typically find is that as your compound becomes more lipophilic, it also becomes more promiscuous. That is, it hits more off targets. So it's a good idea not to have your drug more lipophilic than it need be. Lipophilicity is readily calculated. There are very good programs out there that provide most of the time pretty good estimates of both log P and log D for drugs. And they do this by calculating the contributions. Log P can be made up of the different fragments of a drug, and you can get a pretty good sum. If you then add in a calculation for the pKa of any ionizable centers, you can generate a log D as well. You can get effects whereby, for example, conformational changes mean that the fragments that you put together for your calculation don't quite add up. So any calculated value for log P should, if it's important, I would say it's then you should get a measurement, but a lot of the time calculations are very useful. Along with lipophilicity, solubility is extremely important. Solubility is typically measured in pH 7.4 buffer. You can use more sophisticated systems if you want to that simulate gastric fluid. There are formulation recipes known as FASIV and FESIVs, simulating the fasted and fed states, respectively. Solubility in buffer depends on two factors. It depends on the lipophilicity of the molecule, i.e. how readily it goes into aqueous systems, and also the crystal packing energy of your compound, so the melting point, which it translates into the melting point. The higher melting your compound is, the more energy has to be put in to break up those crystal structures, and therefore the less soluble it is. So there's a general solubility equation that's been generated, which gives this formula where you multiply, there's a sort of intrinsic solubility, and then you multiply that by the melting point minus 25, melting point of degree C, and subtract the lipophilicity. So the higher the lipophilicity, the higher melting, the less soluble the compound is. Solubility, therefore, because of this crystal packing energy, depends on the solid form of the material. So if you've purified your molecule by HPLC, as you may well be doing, you might find that your initial amounts of material are amorphous. They will be more soluble than crystalline material. And so knowing what your solid form is relevant when you're measuring solubility, and particularly because a lot of the time you will end up with the most stable form as being what you dose, or potentially even if you don't dose it, it will form within the patient. So understanding solubility is very important because solubility will affect your bioavailability. Now, one of the reasons you look potentially at simulated gastric fluid is that the contents of gut, things like bile acids, can affect solubility and make compounds more soluble. There are calculations available for solubility. My experience is they're not as accurate as the calculations for lipophilicity. Nevertheless, I think they are still useful to do, and they're on the whole computationally very cheap. So it's something that you can do along with calculating lipophilicity ahead of synthesis for all compounds. Solubility, as I've said, is particularly important in terms of absorption from the gut because only compound that's dissolved can be absorbed. And this is particularly important when you're doing your safety studies, which have to be run at higher concentrations than you will dose to patients. So even if you had enough solubility to be able to administer your desired dose, you might still have a problem for a safety study. So solubility, something that med chemists need to be aware of. Right, the lead identification phase. So at this stage, you've got some hits from your screening process probably, and you've got some kind of assay cascade in place, maybe not everything, but you've got enough to start a program. And so you start your medicinal chemistry design, make, test and analyze. You've got multiple series that you're going to be following up, and you're going to want to try and do that quickly. You'll synthesize analogues of these to build a picture, try and understand the structure-activity relationships. You'll use library, parallel synthesis wherever possible. And what you want to find out from this is which of these series can be optimized. Do they really have opportunities for potency? What's the metabolic stability like of these series? Once you start getting some good compounds, what are the safety profiles like? Are the issues that you find with them things that can be addressed? And as you make progress, you'll start taking compounds further through the testing cascade. And the objective of lead identification is to find one or more series that have the potential to become candidate drugs. So what you want at the end of a lead identification is compounds with confirmed activity at the desired target and where you believe there are opportunities to improve probably both potency and pharmacokinetics, and where the safety risks appear to be manageable. Ideally, you'd like to be able to demonstrate pharmacodynamics, that is, an effect in vivo in an animal model with a compound from at least one series, ideally probably one compound from each series you want to take forward. And the point of this is that you are building confidence in the mechanism that you have, in the chemical equity that you have. So as an example of hit-to-lead chemistry, I've selected an IKK2 inhibitor program that was run by some colleagues of mine at AstraZeneca a few years ago. IKK2 is a kinase enzyme. It's involved in the immune response, and inhibition was hypothesized to be applicable for the treatment of inflammation in chronic obstructive pulmonary disease. So amongst the hits from high throughput screening were these two compounds I've called 1 and 2, and on the left hand side I've got some assays and the generic criteria that were there at the time for what was needed to be a lead. So this is where the project wanted to get to. So they wanted to have activity at an enzyme of less than 0.1 micromolar. They wanted cell activity of less than 0.3 micromolar. They wanted rat hepatocyte clearance of less than 14 microliters per minute for a million cells, and human microsome clearance of intrinsic clearance of less than 23 microliters per minute per milligram. They were looking for a molecular weight of a sort of reasonable amount, so less than 450, and lipophilicity in terms of log D of less than 3. And I've coloured these results in sort of traffic light effects, although I've got two intermediate stages. So green is where they're good, yellow is where they're not far off being good, orange is perhaps not quite so good, and red is bad. So both of these compounds there where are they? They're sort of 16 and 20 times less active at the enzyme level than the target was. The cell activity, this one is quite a bit worse. That's probably because it's not very lipophilic. Normally, you need to have a more lipophilic compound to be able to get into cells. And you can see this compound here, actually the drop-off from the enzymes, what, about fourfold, isn't it? So, yeah, that's okay. This one, on this case, the rat hepatocyte clearance is just about at the borderline. This one's not much worse. Human microsomes there are a little bit poorer, a bit above the target range. But yeah, molecular weight's good. Log D of this one looks really nice. So, well, these two compounds look a bit similar, so they combined them. And they made this compound, compound 3, and suddenly, wow, potency looks great. Cell potency, yeah, we're not far off actually our target. It's a bit more of a drop-off. It's now nearly a tenfold drop-off, isn't it, from the enzyme activity. And the rat hepatocyte clearance got a bit worse. Human microsomes, not that great. Still got nice molecular weight and log D. Well, let's replace the phenyl, the thiophene with a phenyl. More potent still. Still about a tenfold drop off to the cell, but this is now below the criteria that was set at the beginning, so that's good. And the rat hepatocyte clearance has come right down, so this is much more stable. Human microsomes, however, a little bit higher, still good here. At this point, the project did really extensive SAR exploration, so they explored all the regions of this molecule. They made replacements for the urea, replacements for the amide, they looked at alternative heterocycles for the thiophene, and they made a huge number of phenyl substitutions. And this led them to the optimized analog with a parafluoro, still very good enzyme potency, cell potency, much the same as the phenyl, but significantly improved metabolism and still good potent, still low molecular weight and reasonable log D, meeting the criteria, and thus enabled the project to go into lead optimization. They did some more work. This compound had, as you would hope, based on the low rat hepatocytes, had good rat pharmacokinetics, so low clearance. It's a neutral molecule, so the volume of distribution of just under 1 litre per kilogram is probably what you'd expect. The half-life is quite respectable in a rat, and that obviously that relates to this clearance. And the bioavailability of 78% is good. This compound therefore provides a nice tool molecule that can be used for studies and demonstrating effects in animal models. So I finish here. Thank you for listening, and I hope you found this an interesting introduction to medicinal chemistry.
Thank you, Matthew, for a great talk. We'll continue the medicinal chemistry crash course in our next lecture as well, so be sure to tune in for that.